Good afternoon, everybody. My name is Ben Chai, and I'm broadcasting from the University of Amsterdam right now. And I would like to welcome you all to the sixth webinar in the webinar series from CompBioMed. I would like to remind you that this is run in collaboration with the VPH Institutes. And with this webinar, we would like to have all the questions held until the end. So what we would like you to do is to write your questions in the question box. I will then read the questions to our speaker at the end of the talk to just try to keep this webinar running as smoothly as possible. We will also have 4 polls throughout the webinar. So you will intermittently get a poll where you can just answer the questions and stay engaged during this webinar. So without further ado, I would like to introduce Professor Andrea Townsend-Nicholson, who is a chair in biochemistry and molecular biology in the Division of Biosciences at University College London. Today, her talk will be on innovations on medical student training. So without further ado, I give the floor to Andrea.
Oh, thank you, Ben. So what I wanted to do in this webinar was to describe for people work that we've been doing in CompBioMed to teach people how to learn more about computational biology. And one of the things that we had identified was that there was a whole category of people who were very interested in learning about computational biology, but they didn't have any computational experience because they came at it from the biological side of things. And one of the things that we wanted to do was help people learn early during their university education so that they could see these techniques put together and use the computational side as well as the biological side in an integrated way. And we started doing this in our science courses and also in our medical school courses. And so what I'd like to talk about today is what we think is a very neat example of how you can quite easily do some experiments and some computational analysis of them using high performance computing. And we have done this as part of the medical school training program here at UCL. So we're just going to start with a first poll because we'd like to understand what people's backgrounds are who are watching this webinar. We'd like to understand whether you're a computational person or you're an experimental person. Maybe you're both, maybe you're neither. So we'd like you to tell us a bit about yourselves. And let's see, so most of the people here are computational, but happily we've got quite a lot of people who do both. So hopefully you'll be able to see both sides of what we're showing here, and we'll be able to have some people who are computational learning a bit more about the biology and vice versa. So let me continue, and I would like to talk about personalized medicine because this is the thing that is most of relevance to many of us, whether we're working in the area of academia or working in the clinical profession, or whether we're working in industry in some way, or whether we're just somebody who would like to see a drug that's made for us rather than being made for someone else. And this is an ideal time now for us to be able to look at personalized medicine and make bespoke solutions for individuals, because supercomputers have gotten to be tremendously powerful, and we have an awful lot of information about different genomes, and it's become very easy to obtain this. So the post-genomic era of biomedical science is allowing us to have information about individual people's genomes, so we can see what makes things go wrong, how one could correct them, and basically build solutions, healthcare solutions for individuals. I'd like to start by showing you a film of what healthcare could look like in the future. This is something that my colleagues and I are trying to build, and hopefully this will help you understand what we're trying to do. We're trying to build a virtual human. So let me show you about the virtual human.
bodies are exquisitely complex machines. In our efforts to care and improve them, we look to those who push the limits of what is humanly possible, such as athletes. Now, you would say, what can we learn from them if every athlete is different? If they achieve peak performance through customized diets, training, and technology? As it turns out, we too are all different. That is to say, there is no single lifestyle, no diet, no medication that will work equally well for all of us. If this is so, how can we get the best out of our bodies? Now, imagine a virtual human, not made of flesh and bone, one made of bits and bytes. And not just any human, but a virtual version of you, accurate at every scale, from the way your heart beats down to the letters of your DNA code. You know that many drugs only work well on some people and can cause serious side effects in others. The reason is variations in DNA, our genetic differences. But we understand how these DNA differences change the building blocks of your body, the proteins, and we can simulate in a computer how drugs interact with them. By testing drugs on your virtual body, your doctor may eventually be able to test a wide range of drugs and select precisely the right one to suit you. But sometimes, choosing the right drug is not enough. We must also be able to deliver it to a precise target in the body. Take inhaled drugs, for instance, which can end up in the walls of your nose or at the bottom of your lungs, depending on the intricacies of your particular respiratory system. Using supercomputer simulations based on scans of your lungs, we can predict with high precision where particles will flow, and then design devices that can deposit drugs exactly where your body needs them. Well, obviously, mastering drug delivery means controlling the body's main transport network, your circulatory system. By simulating the movement of red blood cells and other cells, we can understand important protective processes, like those that prevent blood loss after an injury. Virtual humans could help doctors to plan risky surgery too. They could be used to work out how to reach an aneurysm deep in the brain that is at risk of rupture, which could cause a stroke. Surgeons could then try out the best treatment or implant to suit the location and shape of that particular aneurysm. They could even double-check that the implant will not cause problems, such as clotting, before they try it out on you. Physics, chemistry, and biology intertwine in your circulatory system to drive the most remarkable pump that evolution has ever created, the heart. To create a virtual heart, processes have to be modeled at multiple scales, from the contraction of muscle to the blood flowing through its chambers, to the movement of charged atoms during a contraction. Virtual hearts already beat within supercomputers, where we can test the effect of different drugs or pacemakers, but also more fundamentally, to understand the way an individual's heart works. Your virtual avatar will not only be built in your image, it will also move like you. We can use it to calculate the forces and the mechanical stresses that are constantly induced on your bones, and predict the risk you will suffer a fracture. Doctors can also use virtual humans to work out how diet, exercise, or drugs can help those with brittle bones. And what if we go even further than treating disease, using virtual humans to prevent illness? Within a supercomputer, many virtual versions of you can explore small changes in your lifestyle and how they affect your health, aging, and quality of life. Virtual humans, helping you to figure out the very best that you can be.
So that is what we are working towards here on CompBioMed, is the ability to be able to prevent as well as treat disease. So we are interested in teaching medical students so that they can see the power of developing a virtual human and the sorts of things that one can do with a virtual human. So I would like to describe for you the kind of work that we have done with the medical students. This has been in the area of metagenomics and microbiomes, and I'd like to give you some biological background so that you understand what we've been doing and you can understand the kinds of analyses that we're looking at with the supercomputers. So what we basically have been doing is looking at the bacteria that are present on the palm of each of our students' hands. And these, what are they indeed, are a number of the different definitions that exist for terms that you might find used in the context of microbes present on your body or on your skin or in some kind of a microbiome. So the overall term of metagenomics is something that relates to the study of genetic materials that are recovered directly from environmental samples. And these don't have to be skin samples, they can be any kind of sample. They can be water samples, they can be soil samples. There are bacteria throughout all the different sorts of things that you might wish to sample and look at. We happen to be looking at the microbiomes present on human skin. Another term that you might see used for this is microbiota. This is the community of microbes that are found on or in multicellular organisms. So we're looking at the skin microbiome, but there are others that are human microbiomes. So this is the microbes, and the microbiome is the genetic material of the microbiota. So that is the individual genomic DNA from each species, which will be unique to it. So in the medical school training that we have been doing, in the education we've been doing, we have called our course From Skin to Metagenomics, You and Your Microbiome. So we go from identifying the microbes on the human skin, that's from the skin part, all the way through to metagenomics, which is identifying what they are based on the genetic material that we have obtained, sequenced, and analyzed computationally. So they're very similar terms, and you might see them used interchangeably. This is a slide that gives you a sense of perspective of metagenomics, the timelines and key important things have happened in it. And you can see from this that we've been studying microbiomes for a very long time, almost 350 years. So 350 years ago, approximately, a little bit less, we've had observations about the microbiota in human mouths. People started being able to isolate microbial cultures, started to do ecology experiments, and then people identified that things that were highly conserved between species, so the same gene in many different species, could be used to look at the small differences from species to species and use them as a marker for the classification. So here we can see bacteria, archaea, and eukarya, these three different classifications. I'll show you a bit more about these in the next slide, but you can use particularly highly conserved genes as markers to be able to see who's related to whom. In the late mid to late 70s, we started to develop DNA sequencing technology, which has moved through several generations. This is the initial kind of chain termination sequencing that was done with radioisotopes or with fluorescent dideoxynucleotides. And you can see that it's moved all the way through to the next generation sequencing technologies. And this graph shows you the relationship between the number of metagenomic publications that scientists have been able to publish and the time down on the x-axis. And as you see the technology increase, you see an increase in the number of publications. And this is something that happens in pretty much any technology you look at. As the technology advances, you see an increase in its uptake. The first people who start to use it are the early adopters, and typically normal average users come along about 15 years later. Another technology that I'd like to, this was a Nobel Prize winning technology, DNA sequencing, as was PCR. And PCR is the ability to a technology by which you can make many copies of a piece of DNA. So we do this in our metagenomics to be able to get enough material to sequence to be able to identify the samples. So these are the two pieces of technology that we're using in our education program. We're using DNA sequencing and PCR. And you can see that as we come up towards the present day, particularly with respect to DNA sequencing, the cost to sequence a human genome has gone from incredibly expensive, this is one single human genome, down to quite inexpensive. So this would have been around the time, 2003, when the human genome was first published. And this green arrow indicates when high throughput sequencing, the next generation sequencers, entered the market. And you can see that the cost dramatically became less expensive. You can see a few wiggles in the lines around here, but around 2015, the cost of sequencing a human genome got to be approximately $1,000 per genome. And around 2015 is when we started using the next generation sequencing in our medical school training program and in our science student training program, because this cost is so inexpensive that you can now use these state-of-the-art technologies in teaching in universities. And so we were one of the first to do this, and we're very pleased to be able to use these technologies for our students. This slide shows you the classifications, starting with life, biota, all the way down to individual species. Our species is sapiens, our genus is Homo, so we are Homo sapiens. And as you go up the classification structure, things that are more like each other get grouped together. You may have learned this classification when there were 4 kingdoms, and these have now been replaced by 3 domains. And you can see the 3 domains here, which are indicated in the relationship to each other. This thin green branch here is the eukaryotes, which includes our cells. We have the archaea here, and then this branch here are the bacteria. So you can see that, relatively speaking, the bacterial domain is huge compared to the other 2 domains. And the prokaryotes, there are about 1 billion prokaryotes, 10 to the 9 prokaryotes. So this is very large indeed. And as we come on to look at our analyses that we've been doing in this medical student innovation, you'll be able to appreciate now some of the constraints that we have in our experimental system. So that's a bit of the background. Let's take a look at skin microbiomes and how we characterize them. We basically go through 4 steps, and I've talked about the technology that's involved in 2 of these. We have to purify, well, obtain the sample and purify the genomic DNA from it. We then have to amplify it using PCR or polymerase chain reaction so that we can get enough material to sequence. We will be sequencing a marker gene, and we will be taking and analyzing this marker gene result using supercomputers. So the first 3 steps here in blue are experimental, and the step in green is computational. So let's start with purification. This is when we obtain our samples. The way in which we will be doing this is to take and have a swab. You can see a swab here. It's a flat cotton surface that we dip in a liquid that has a little bit of soap in it, but not too much. And we basically wipe this across the palm of the hand repeatedly on both sides of the swab until the sample has been obtained from the palm of the hand. And then we pop it into the swab into a test tube, and it snaps off quite neatly so that all of our purification steps afterwards can be done just with the microbes that are on this little swab head. And we're collecting the microbes so that we can purify their genomic DNA, and their genomic DNA is unique to each species, and this is what we use to identify what is in the sample. This is a slide that I've taken from the National Human Genome Research Institute that shows the different sites from which you can obtain skin microbiomes and some information about the properties of the site. You can see the characteristic of the site has been identified as whether it's oily or moist or dry. You can see from the front of the body the different sites that you can sample, the back of the body, the different sites, and you can see some preliminary information that they've given us here about the different kinds of things that they found on these sites. This is where we've collected. This is the palm and the palm of the right hand. This is a dry site. And the reason why we collected from the palm of the right hand, we call this the handshake experiment, because when you meet somebody, you shake right hands with them. So this right hand is what contains the microbiome that you most frequently present to the world and that the world presents to you. So we thought it would be interesting to look and see what was on this microbiome. So this is how we collect our sample. This is what we are going to be looking at in our sample. This is a piece of human genomic DNA. I know we're talking about microbes, but I've given an example of a piece of human genomic DNA. And in this piece of genomic DNA, we have a number of ribosomal RNA genes. Ribosomal RNAs are not translated into protein, but they're very important for protein translation because they are part of the ribosome, which is the protein-making machinery. And the ribosome reads the messenger RNA and makes protein. And we can see that you can have prokaryotic ribosomes and you can have eukaryotic ribosomes. They have large subunits and small subunits, and these are made up of RNAs as well as a large number of different proteins. And you can see that as a general rule, the prokaryotic version is always slightly smaller than the eukaryotic version because eukaryotes are a little bit more complex than prokaryotes. And you can see here that the small subunit of the ribosomal RNA in prokaryotes has the small subunit of the ribosome rather has a 16S ribosomal RNA in it, whereas in prokaryotes it has this 18S ribosomal RNA. So the 16S ribosomal RNA is very important for protein synthesis. This is the RNA that brings the messenger RNA that's being translated into the ribosome. And why it is a good marker gene to use for us is because it is unique. The 16S ribosomal RNA gene is unique to bacteria, to prokaryotes. It doesn't exist in humans. Humans and other eukaryotes have an 18S. So we can do an experiment that only detects microbial DNA and doesn't detect any human DNA. The next stage that we have in our experimental protocol is to amplify. We have obtained our sample, we have purified the genomic DNA from it, and now we need to make enough of this DNA to be able to sequence, and this is where polymerase chain reaction is going to come in. And so this is a diagram to explain to you how you can make many copies of the same region of DNA using polymerase chain reaction. Your DNA template, which is double-stranded, is going to go through 3 separate steps: a high temperature step, a mid temperature step, and then a slightly higher but not as high as the first temperature step. And 3 separate things happen in these stages. In the first, the temperature is high enough that the hydrogen bonds holding the DNA helix together are broken and the 2 strands separate. We need the 2 strands to be separate because we are going to make very specific small pieces of DNA that will prime the synthesis of this new strand here. So we go from one copy that's double-stranded to two copies that's double-stranded by the time we finish this third step. The temperature of this second step is low enough to allow these primers to anneal to their template, and the temperature of this third step is high enough that the polymerase can continue the synthesis of the strand. And when we finish this, we have two copies where we started with one. And we repeat this through a number of different cycles, so we can amplify 2 to the n times, where n is the number of cycles that we perform. And when we have enough cycles performed, we have enough DNA that we're able to detect it and sequence it. So I was talking about the 16S ribosomal RNA gene, and this is what the 16S ribosomal RNA looks like. It's an RNA molecule, so this is its 5' end, and this is its 3' end over here. It has a great deal of secondary structure within it. This is shape that comes about from intramolecular hydrogen bonding within the 16S ribosomal RNA itself, so you get all of these stem and loop structures, and you can see indicated in colors these different regions, V1, where's V2 gone? V2, V3, V4, V5, V6, V7, V8, and V9. And these V regions are variable regions. So if we look at this graph here, where we have on the x-axis the nucleotide position, this is towards the 5' end here. This would be the 50 here, all the way out towards the 3' end. So this is going to be where 1350 is, right here. That's the end of what we see on this graph. The higher the H', the more variability there is. And so you can see that the highest bits of variability are corresponding to these V regions. And there are also regions where there's very little variability at all, where the H' value is quite low. And these are very constant regions where there's a great deal of conservation of identity if you compare different species. And so we use these gray regions here to design the primers for PCR that I showed you here. So these are the gray region primers. And we use this to amplify the hypervariable 4 region of the 16S ribosomal DNA. So we are making many copies of this small piece of DNA back and forth there. So let's talk about the marker gene sequencing. We know that we're sequencing the 16S ribosomal RNA gene. So this is our marker gene. And I've made an excerpt here to show you what this V4 region looks like. This is its secondary structure in the 16S ribosomal RNA gene. Here we have it drawn out in a linear fashion from its 5' end to its 3' end. And we can see here 515F. This is the forward primer for the PCR that we're using, which starts about here in 515. And we have this other conserved region, 806 is the reverse primer. It's a little bit off on this side, and we are using it to amplify from this region here all the way around and through and a little bit beyond to this region there. So we do this with polymerase chain reaction and with very specific primers. So the primers have a region that is going to be able to hydrogen bond or anneal with the 16S ribosomal RNA gene. And it also has regions outside of it that relate to the DNA sequencing, including this particular barcode. So this first region, these primers are for PCR, and then the region that's amplified, we will be making a sequence run of, and these are the primers that relate to the sequencing run. So the primer barcodes, this barcode here, which I've showed you, and we can see that it's in red there. Here's our red barcode. The primer barcodes allow us to take a look at the different samples. And so you can mix all of the samples together onto one DNA sequencing chip, and the barcodes will allow you to tell which sequences come from the red sample, which sequences come from the blue sample, which sequences come from the green, and which come from the purple. So when our students collect their samples, each is given a barcode that is unique to them and to their samples, so they know what bacterial species are present in that. These are all mixed together. They go onto the sequencer. We use Illumina sequencing technology. We use either MiSeq or HiSeq technology, depending on the number of reads, which is the number of sequences we wish to make. And then we obtain back these individual sequences that correspond with the barcode to what is present in the sample. We then go through data processing and quality control, which is something that's done computationally, and then we are able computationally to understand more about our microbial community. So this is the process experimentally. So I think it's time now for our second poll, where we are going to take a look and see, for those of you who are computational scientists, do you think you'd be interested in learning experimental techniques that relate to your work? And if you would, what kind of a thing might you be interested in doing? A short workshop or something a little bit longer? And if you're not a computational scientist, you can either fill in as you like or don't have to submit. Okay, so we can see, I can see here that there are a number of people who would be very interested in attending a shortish workshop and a number who would interested in obtaining something longer. So you might be interested in doing this skin microbiome practical if it's something that takes your fancy. So let me hide that, make that go away, and we shall move on. So this brings me, now that we have taken a look at the experimental side of things and understood the biology behind how we've designed our experiment and the kinds of things we're sequencing, this lets me talk a little bit more about why these are innovations in high performance computing training and for medical students and science students and engineering students. So CompBioMed is a center of excellence that is EU funded and designed to be focused on users. And one of the things that we are particularly keen in doing is bridging this computational experimental divide. You can see in what I've described to you as an experiment that in order to understand what microbes are present on your skin, on the skin of your hand, you need to be able to do both computational and experimental work. So CompBioMed supports existing users who need these technologies, the computational analysis resources, and develops existing users. So for example, those of you who are computational, we can find ways of providing you with support if you'd like to learn more about the experimental side of things. But we're particularly taken by the fact, and we don't think that this has been done before, which is why we think it's an innovation, that we can create a new category of users of high performance computing, future users. And these are people who will be as fluid computationally as they are experimentally. And these courses that we are running and the course that I'm describing here will help to generate these future users. And of course, because this course is up and running and people have been using it, we can make all of our educational templates and our expertise available to anybody else who'd be interested in adopting it, and we can help you do that. In fact, from 2019, our renewed Center of Excellence is going to be focused on doing exactly this in medical schools in the University of Amsterdam, which is partnered by its HPC facility, national facility, Sir Sarah, at the universities of Sheffield and Oxford, and at the Universidad Pompeu Fabra's Medical School in conjunction with the Barcelona Supercomputing Center, which is the Spanish HPC facility. So this is the thing that we were running. You can see it's a student-selected component. What a student-selected component is student choice in their curriculum. So many people in medical schools have to take a defined curriculum, and then there are elements that they may wish to pursue in their own areas of interest, and it's done at UCL through these student-selected components. And so these are first-year medical students in the labs and in computational practicals, doing this from skin to metagenomics, exploring your microbiome. So we started with science students in 2015, we started with the medical students in 2017, and this is my colleague, Dr. Pringle from the EPCC in Edinburgh, describing for the medical students the machine that they will be using for their particular computational analyses. And this is my colleague, Dr. Martin, who is at UCL and is helping people to understand a little bit more about the computational side of things so that they're comfortable using these HPC resources. So I'd like to describe for you the experimental workflow that we've done, then the computational work that we've done, and then end up with where we wish to go from here. So this is the workflow that these students have followed. This is them doing it. This is where I showed you about the sample collection with the swabs. They then isolate the genomic DNA, take a look on a gel to visualize it on a gel to make sure that it's of a size and an integrity, not broken up, not damaged, so it's able to be used for PCR. This is when they do the PCR step to amplify their marker gene for sequencing. So this is the hypervariable 4 region of the 16S ribosomal DNA. The PCR gets checked on the gel, and then the fragment that's been amplified is purified. We quantify it, and then because everybody's samples are going on the same chip, we get the concentrations equal to each other so that each sample is represented equivalently across the chip. So we pool the samples at the same concentration. We send them out to our colleagues at UCL Genomics. Well, we don't send them out. We walk them across the road to UCL Genomics, who put this on their Aluminum iSeq machine, and then we get back from them the data for data analysis. So this is the experimental workflow that people carry out. And that brings us to the end of the experimental side of things. This blue box now with the red data analysis in it is moving over to the computational side of things. And now we're going to have our third poll for you experimental people out there. Whether you'd be interested, we'd like to know if you'd be interested in enhancing your computational biology skills. And of course, you have an option that's not available to people who wish to learn experimental skills, because those are not something that can be done online. Those are a hands-on thing, whereas the computational side of things you might wish to do online. So there's a lot of interest in doing an online course, and some reasonable interest in attending a training course or a workshop. So we shall see how we can meet people's needs and their interests in that area. So I'm going to move now towards the end of this webinar, towards the computational analyses and the DNA sequence analysis that's being done of the 16S ribosomal rDNA that come back. And I've simply, rather than making complex computational workflows for this, I've described the main steps that we had to do. And the first of these was something that I was very grateful for the support of my colleagues at the EPCC, at Surf-Sara, and at the Barcelona Supercomputing Center, because we needed to take and port the computer program CHIME that we use to look at microbial diversity onto HPC architectures. And so we ported CHIME onto HPC machines at each of the 3 facilities that are core partners in CompBioMed. And we learned an awful lot about similarities and differences in the different architectures, and also that we were able to, one way or another, make a program that had never been designed to work on high-performance computing machines before work very well. So my colleagues are absolutely fantastic in helping with that one, and I owe them a great deal of gratitude for that. The other thing that we had to do, if you remember, most of our students are in their first year of medical school. They're not particularly familiar with computational. They have more experience of experimental work than they do of computational work. And many of them are used to touch screens, iPhones and Android phones and tablets and the like. So we had to spend some significant amount of time teaching people who'd never seen the command line before how to use the command line to do the sorts of instructions that they were used to doing with a mouse or with some other form of communication on their devices. We also needed to introduce people who'd never used supercomputers before to high performance computing, editing files, running batches and those sorts of things. So we had some computational training in there once QIIME was up and ready to be used, and once we had our users ready to use QIIME on an HPC architecture, then we were able to go on to data analysis and use QIIME on the different machines that they were given access to. So this is the starting point where they start from their sequencing data, and they go through different steps using QIIME to work out the richness of their species and come out with plots where you can see relatedness of samples. And this is just an indication of a PCOA plot, a principal coordinate analysis plot. So all of our students took their sequencing data, the metadata associated with it, and were able at the end to generate these principal coordinate analysis plots. So the results of the work that we did in this SSC enabled people to identify which bacteria and archaea are present. Remember, these are the species that have the 16S ribosomal RNA genes. We were not able to see eukaryotic species because they don't have 16S ribosomal RNA genes. We also could see which were present at significant representation. Now, if you remember, I described at the start of this webinar that there are over a billion different species of prokaryotes. So if you sequenced 200,000 sequences in your sample, if your sequencing depth was 200,000, you still aren't in a position where you would be able to see whether you could detect one of a billion prokaryotes. So we cannot sequence in the depth needed to say that an individual species is present or absent. We can only talk about significant representation. And our success rate, because we had 135 students doing this experimental and computational work, was 100%. So all 135 students were able to use the high performance computers and QIIME on those architectures to be able to conduct their analyses and obtain their results. So these are some of the caveats. I mentioned that you can't really see at the level of individual species because of the sequencing depth we do. So you can definitely see phyla, which is what most of the software will bring back for you. You can see some greater level of detail than that, but you can't see individual species. This is partially because of sequencing depth. It's also because we're sequencing a single gene and not a whole genome. You would get better granularity if you did this with a whole genome. And not only are we sequencing a single gene, we're sequencing a small region of a single gene. So we're sequencing approximately a sixth or a seventh of the total size of this 16S ribosomal RNA gene. So now it's time for our fourth poll to find out, after having followed what we've been speaking about in this webinar, whether you would be interested in coming and doing a You and Your Microbiome hands-on experience where you get to do both the experimental side and the computational side to find out what's on your right hand as far as microbial species are concerned. So it sounds like that might be a possibility for people if they're interested. So we might be able to use your support for this and offer microbiome summer schools to have a bit of a play with the experimental and the computational side. So I said earlier that from 2019, we will be adopting the CompBioMed educational expertise across medical schools in the EU, and I mentioned the 4 that we're starting with. We're not restricted to these in any way, so I just wanted to very briefly here find out whether participants who are either live with us now or seeing this as a recording afterwards, because this will go up on the CompBioMed website, see whether people would be interested in running this kind of a computational experimental course at their own institutions or their own universities. So we have been running this now in one version or another for about 3 years, and live in the medical school curriculum at UCL for 2 years. And we have it down to quite an effective workflow that can be done either as a 1-week workshop or can be spread out across a term, but basically can be encapsulated in 24 or 48 contact hours. And the laboratory side of things is basically 8 to 10 hours worth of work, and the rest of the time is on learning how to use HPC and learning how to work with QIIME and the data analysis. And these are the components of the modules that we can help you set up if you're interested in doing that, and help you run until you get a feel for it. So introductions, the students design their own protocols for the kits that we have identified that are best to use. We introduce people to next generation sequencing so they understand how the barcodes work, and they have workshops in computing and high performance computing. Then we go through the experimental steps, and then we go through the data analysis and the comparison of data and the summary of findings. So I'd like to acknowledge all of the core partners in CompBioMed who were instrumental in helping us with the development and the running of this program. I mentioned my colleagues at EPCC, SURSAR, and BSc, who were hugely helpful in getting QIIME up and running and working on their supercomputers, and colleagues at UCL who helped run this module, and colleagues in the medical school who were able to help us get support for putting this module into the curriculum, and my colleagues at UVA who are helping me run this webinar and describing for you what we've been doing. So I would like to turn now, Ben, I think, to a question and answer session, and we can see if people have questions that they would like to ask about what's being presented here.
Yeah, thank you very much, Andrea, for a great webinar. We have 2 questions at the moment.
Okay.
I will read the first one. It's from Andrew Narracott, and he asks, what are the rough timescales for the relative stages of the process, i.e. time to undertake experimental analysis and time to run computational analysis?
So the experimental analysis is something that these steps can be done in approximately 8 hours. And when we do it, we run over 2 weeks at 3 hours a week. So we've done it in 6. And sometimes it's nice to have a little more time to get the right number of PCR cycles. So we've added a third, so that's 8 or 9 hours. We send the sequencing out to a sequencing facility that has Illumina sequencing, and that can take some time to come back. So we usually program that in advance, and we warn our facility when we are expecting to have samples ready for them, and we basically book onto their machine. I think we get samples back within a few days. So the experimental is probably 8 hours in the lab, a week waiting for the data, and then as long as people like to analyze the data, how much time you spend on the computational side depends on what people's backgrounds are, but we have done maybe 15, 12 hours of workshops with students on the computational side of things, and that's included their analysis. And we've also helped them understand the way in which you can do statistical analysis, which is a little bit different from the computing. So experiment 8, a week to wait for sequencing, 12 to 15 of computational would probably be a good breakdown, and you could expand any of those areas as you like.
Thank you for such an in-depth question. We do have one more question from Andreas Kuko. He asks, HPC resources are normally reserved for intensive research-type computing. How did you manage to get 135 students using HPC resources? It must have caused some resentment among other colleagues.
I'm not aware of any resentment. It was quite easy to do in some ways. It was easier to get access to the resources than it was for our students to use them in one way. there is training accounts time for each of these facilities that goes with the grant, the CompBioMed Center of Excellence Award. And so we were using this training account. There are difficulties when you put new users onto systems like this. We had everybody in a student cluster. Why I say it was more difficult for them to use the machines than to get access to them is because we discovered in the student cluster that if students mistyped their password, which was something that they were quite likely to do because they are less familiar with typing passwords over and over, and some of the keyboard letters like a 1 and an L look similar to each other, they would lock themselves out of the system quite frequently. So on one of the facilities, we had to whitelist their access to make it easier for them to get on. And then we learned a bit about how our networks were constructed and how our IP addresses were constructed. So it wasn't just one person that was having troubles repeatedly. So we had some issues like that. But once that got workable and everybody understood about that, we were perfectly fine. There is a larger question here about availability of resources. And what we are hoping is that as more people learn about the power of computational biology and the fact that they can do much of this themselves, then it will provide support for our colleagues in the HPC facilities to make business cases, to make more machines available, and to have machines that meet the different categories of users who are starting to come. We picked a relatively simple, computationally simple workflow that could be achieved in about an hour. We have not yet moved on to simulations, molecular dynamic simulations or things like that. And those are things that would be more time intensive.
Okay, thank you for your answer. Oh, we get we get another question here. It is also from Andreas. Would it be feasible to do the analysis on local workstations?
Oh yeah, absolutely. Chime was originally something that was a piece of software that was built to do on local stations. If there are a lot of machines that are 12 core, 8 core now that could do this sort of a thing. But our purpose was to help people become familiar with HPC and to have an entry point into understanding how to use it so that they could do more computational things. So, for example, with our colleagues at the University Pompeu Fabra and the Barcelona Supercomputing Center, we're developing new SSCs that will look at heart modeling, like you saw in the virtual human, to know where to position pacemakers. And the idea would be that a clinician in the clinic who needs to put a pacemaker into a patient would be able to look at that patient's virtual human, take the image of the specific patient's heart that has been obtained through imaging methodologies, and use a computer simulation to work out where best to place a pacemaker to rectify the cardiac rhythm for that patient. And that's something that is going to be much more computationally intensive than what we've been doing. We chose something to introduce our medical students to the idea that they could have access to this kind of compute for helping to treat patients when they are graduating and practicing as clinicians.
Okay, thank you very much. At this moment, there are no further questions. So I would like to thank you once again, Andrea, for a great talk and great questions. I would like to remind everybody that the slides and the audio from today's webinar will be made available on the CompBioMed website. Also, a finer point about the video, which was shown at the beginning of this webinar, maybe wasn't as high resolution through this webinar interface. If you want to look at this video, it is also available on the CompBioMed website. So with this concludes the 6th webinar of the CompBioMed webinar series. And thank you all for joining and participating.
Thanks, Ben.